Hey everybody, how's it going? So we're gonna talk about one more topic. This is about gene mutations. So right now we've been talking sort of like the ideal world, everything works out perfectly. But it doesn't necessarily work out perfectly. Sometimes our genes get mutated. And one of the ways that this happens is from substitution. So what that is one of these letters might accidentally get transcribed incorrectly, which would lead to a different amino acid and a different protein being produced. So we're just going to go through some examples and sort of explain the process that way. I started with a DNA strand here, and I am just going to transcribe it to RNA, and we'll see what we get. So our T becomes A is U, C becomes G, A, C, G, C, U, G, G, U, G, C, U, G, A. So that would be our RNA that we get from the DNA. More specifically, that would be our mRNA. And then we can take a look at our chart and see what the different amino acids are. So AUG is methionine. AAC is asparagine. CGC is arginine. UGG is tryptophan. UGC is cysteine. And UGA is our stop codon. So there's our RNA, and here are our amino acids. Remember, the methionine is our start amino acid, and here's our stop amino acid with all of these amino acids in between that will make up a protein. So what happens when we substitute one? Let's take a look. All right, so here we have a strand of our mutated DNA. If we compare it to the original one, the only thing that changed was instead of a G, we substituted it into an A. So let's see what happens to our RNA sequence in this case. So our RNA sequence is still going to start with AUG, but now for TTA, it's going to become AAU instead of AAC. And that's the only change that I made. So I'm just going to write in the rest of the RNA sequence from before. And that means that our amino acids, it's still going to start with our methionine, but now AAU codes for asparagine. Oop, there we go. And nothing else changed in our amino acids. So that would be our sequence. Now if we compare it to what we had, well actually, the change in letter didn't change the amino acid sequence at all. We still have the same one. And that's because, remember, the same or different combinations of our codons, they can still result in the same amino acids. So in this case, it is what is called a silent mutation. So yes, the letter was changed, but our amino acids still stayed the same and it still produced the same protein. But we're going to take a look and see that that's not always going to be the case. So let's check another example. Okay, so I changed the spot of mutation. So I put a G back there. And now, instead of an A here, I put a T here. And let's see what kind of mutation happens. So our RNA will mostly be the same. AUG, AAC, CGC, UGG. Now this one will be AGC, and this will be UGA. So the amino acid, still methionine, still asparagine, arginine, tryptophan. And now let's take a look at what AGC codes for. So AGC codes for serine, and then our last one is our stop one. So if we compare it, everything matches up until we get to this point here right before the stop signal. We have a different amino acid. So that is going to code for a different protein. And this is called, well, it's called like a missense basically. So it results in a different amino acid being produced, and because a different one is produced, a different protein is going to be produced. But as you can see, it resulted in a different amino acid. So that's why it's a mutation. We're going to take a look at one more example and we'll see what happens. So here's the third example. The thing that I changed this time was, instead of here it being ACC, here I changed it to ACT. So I already kept the rest of the sequence here. Now ACT transcribes to UGA. And when we look in the table here, UGA is a stop codon. So what that means is this amino acid chain gets stopped prematurely. This could also be referred to as like nonsense. What happened is, instead of this protein being created from the amino acids, we have this much shorter protein, which definitely is not what should be the outcome of the amino acid sequence. Like, we should be getting this protein here. Instead, we're getting this one, and it stops prematurely. Like, we don't even have to consider these other codons because there's no start codon here. It just stops at that point. So yeah, that's called a premature stop sequence. So those are the three different outcomes. Sometimes the outcome won't affect it at all. Sometimes the outcome will change it to a different protein, and sometimes it will just make it stop very quickly, or quicker than it should. So if you guys have any questions about any of this, just let me know. There's going to be an assignment that goes along with this, the same idea. But yeah, if you have any questions, let me know. Have a good one, guys.